Hello, so this is Sarah Sheffield. This is a recording of a conference talk recently given at the NAPC conference in Riverside, California in 2019. This talk was part of the Two to Tango session, which advocates for professional relationships between avocational paleontologists and career paleontologists and understanding the benefits that both get when working together. So, without further ado, I will talk to you a little bit about advances in echinoderm paleobiology, collaborations with avocational paleontologists. So, myself, I am a professional paleontologist. I work at the University of South Florida, and with me on this abstract and presentation are multiple avocational paleontologists who have worked closely with echinoderm paleontologists in the past few years. So before I get started, I'd like to acknowledge a couple groups. So my first group is the My Fossil Project, who sponsored the funding to allow me to give this talk at the conference in NAPC in Riverside, along with the Dry Dredgers and the Association of Applied Paleontological Sciences, which are two avocational groups that have funded my echinoderm paleobiology research in the past. So to tell you a little bit about the backstory, the Dry Dredgers are one of the oldest continuously operating fossil clubs in the United States. They started at about 1942, and the reason they're continuously operating has probably a lot to do with the fact that Ohio is just a treasure trove of fossils, specifically beautiful Paleozoic fossils. So the Ordovician, the Silurian, Devonian, just beautifully preserved in this strata, giving no end to the amount of collecting that you can do. So I became involved with the Dry Dredgers a couple years ago when they awarded some research money to me to fund my dissertation work. So here are some images of the meeting. These are all taken by the official Dry Dredgers photographer. Here on the left we see Tom Bantel, and we see some people collecting fossils and looking at different fossils. Here I am down here in the bottom right discussing some work with a collaborator about some research. And here's an image of the talk that I gave. So I got involved with them in this way, but during this meeting, I actually met with a number of paleontologists, most of whom were on this abstract, that gave me some ideas about further research collaborations. So the benefits of collaboration, before I get into this, before we go any further into talking about the research that has happened from this, is that a lot of avocational paleontologists have an expertise in the field area that just cannot be matched. So avocational paleontologists are able to go to a lot of these localities on multiple occasions, and they're able to look more frequently and year-round, giving a better and broader view of the area which collect. And this gives us a much larger and better collection of specimens to work with, which will be really important for our which is really important for especially echinoderm paleontology, and I'll explain why in just a moment. So here is an image of something called Holocystites. It is an echinoderm that lived in the Silurian, so quite a few 100 million years ago, and it's absolutely beautiful. It's also an incredibly rare specimen. And the reason for that is echinoderms are wildly difficult to preserve in the fossil record. But we'll talk about that in just a second. So here, just to refresh anybody who may not remember a whole lot about this, here is our echinoderm phylogenetic tree. These are all the echinoderms that have ever lived. And as you can see here, we've got the modern groups that you might know a little bit better, like the starfish, the brittle stars, the sea urchins, sea cucumbers. And all the way over here, we have a crinoid. So as you can see, crinoids are pretty far removed from the rest of the still living crinoids today, and they're more closely linked with the extinct crinoids. So I'm mostly focused on the extinct crinoids over here, but that's what we're going to be looking at today, those types of fossils. And specifically, we're going to be looking at blastozoans. And this is a group of echinoderms with special feeding plates. The feeding plates usually come off of here, and they act to catch food to help the organism survive. And they're defined by their respiratory structures. We don't need to know much more about them than that, but it always helps. So if you look here, you see all these little holes all over the body. Those are the diplopore respiratory structures. For the purposes of this talk, we don't need to learn much more than that, but those are the types of organisms we're looking for. And they're incredibly rare. These diploporans are only found in a couple areas on Earth, and usually they're not found in a very high number, which limits what we can know about them. However, in a couple places in the world, that's not the case, and they're found in a really high number. And one of those places is in southeastern Indiana, in what we call the Napoleon Quarry. So here I am at the Napoleon Quarry with a couple of colleagues. Here on the left is my dissertation advisor, Colin Sumrall, and here on the right is an undergraduate research student, Sarah Johnson, working with us at the time, and I am in the middle. So one of the issues with echinoderms is that their taphonomy, so how they're preserved after death, can be tricky. Usually, most of the echinoderms we're going to be looking at are disarticulated within 1 day to a year. So that really lowers our expectations for things being fossilized for hundreds of millions of years. Very few echinoderms end up ever being fossilized, which means if we're already finding them in a very low number, that's going to limit how much we're able to know. So here's what I'm talking about. Even if they're preserved, sometimes they don't preserve in very nice quality. Okay, so if we look here on the left and the right, these are two different specimens. So we've got these two here. This is Holostytes wincelli, and on the right we've got Polycystus sparsus. This is what a typical echinoderm fossil can look like. And you're looking at it thinking, well, that is just an ugly blob. And you may be correct, but it is actually a fossil. There's no plate differentiation. There's no real detail that we can see. But if we look here on the right, you can see all kinds of preservational details. We can see the mouth, we can see the brachial plates laying across us, we can see the anus right here, we can see all these different features. But these are, again, quite rare. And even though on this one, on Polycystus, we can see a whole bunch of detail, there's still a lot that's missing. For example, there should be a number of plates branching off of the arms right here. There should be cover plates over the mouth and over the anus. And those are, even in the best specimen, almost always lost. So this is where the dry dredgers really came into play. With a massive amount of specimens that have been collected over decades, the rarest of these fossils have been found, and that is fossils that still have the incredibly delicate plates preserved. So here's an example of Holocystites with one of the only known representatives that have the cover plates covering the mouth hole attached. Here's another one that's even more beautiful, and look at this right here. This may not look like much, but here is our anal pyramid right here, and it has a series of plates covering it. This is the only known specimen out of thousands that has actually been preserved, these delicate little plates. And here we have one, and one of the only ones known not just from Diploparida, or excuse me, not from this group right here, but from all of Diploparida with the brachial plates attached. So this is a specimen. It's closely related to Triomera. We're not entirely sure who or to what group it belongs, but we've got the body right here, and what we have here is a brachial plate branching off and acting as like an arm structure. And this is an incredibly rare opportunity to see how these specimens actually looked. These are some of the only example in thousands. And this is going to bring me to a second project that I'm working with avocational paleontologists, and that is understanding the growth trajectory of diploporan blastozoans, just like the ones we've been looking at in the previous few pictures. So this is not a diploporan. These here are blastoids. They're closely related, but they're not in the same group. And we're looking right here at a growth series. We have our adults here, the largest, and the juvenile here, the smallest. To understand how these echinoderms are growing, there's a couple things we need. We need an incredibly large sample size, we need lots of representatives, and we need representatives from different stages in life, so small and big ones. But with diploporans, it's pretty rare, as we already know, to have large collections outside of a couple places in the world. But what's even weirder is we need them from different sizes, and holocystids, or diploporans in general, don't have those different sizes. They're mostly all about the same size. We don't know much about their juvenile stages. So here we are actually looking for smaller pieces, right? This was not a fun task, I'll say. It was about 100 degrees that day, but there we go. We didn't find anything. So one thing to note here is that after we left this quarry, and here we have we're looking for places with disarticulated plates so we can find where to potentially scout for more juvenile pieces altogether. So we actually collected hundreds of pounds of sediment to bring back to the lab and analyze. And after many hours of microscope work and many undergraduate researchers, we found absolutely nothing. No juveniles, no disarticulated plates of juveniles, neither a hide nor hair of them. and we don't yet know why. That's a research question for the future. But with an extensive amount of collecting by Tom Bantel, we have found the smallest known specimens of Holocystites, that group that we've been talking about. Now, these aren't the most juvenile we could have, but they're significantly smaller than what we're used to seeing. So we're actually able to put these in a growth study. So with the help of Tom Bantel, we actually now have a study of diploporite and blastozoans involving hundreds of these specimens, and we can begin to answer some questions that have never been asked before about these fossils. So the last project I'm going to talk to you about today is the changing of our biogeographic interpretations. This is a beautiful specimen called Eucystus, and Eucystus lived from the Ordovician to the Devonian, which for a Diploporan, that's a pretty long time. Most of them only live a couple tens of millions of years usually. This one lived much longer than that. So it also has a very wide biogeographic range, found in parts of Asia, found in northern Africa and Europe, lots of different places. Recently, Bruce Gibson, an avocational paleontologist, found a Eucystus specimen in the Bainbridge Formation of the Silurian rocks of Missouri. So this completely changes what we thought we knew about Eucystus. Never before have we found it in North American rock, and now we have it. So that completely changes what we thought we knew about the biogeographic patterns of this particular organism. So the contributions really don't just stop here. So avocational paleontologists within the dry dredgers have actually aided in a number of formal and informal research endeavors. So here is an example of a paper recently published by James Tomka, Carl Brett, Tom Bantel, Allison Young, and Don Bissett. And Tom and Don are also on this abstract, and avocational paleontologists. And here is another example. Here is a recent abstract published at the Geological Society of America meeting, where I, along with collaborators and Tom Bantel, are working together to understand the growth trajectories of a lot of these organisms. But the dry dredgers have really done a lot more than that. If you look at the broader range of their contributions, they've actually funded a significant amount of research. So these are the papers that I've published recently that have been funded, at least in no small part, towards by the dry dredgers themselves. So all of this research has been done because of them. And just in the echinoderm workers, I actually counted it up, but we have a number of 8. We have 8 echinoderm workers in recent years that have been funded through this project, and each of them has published multiple papers using the funds that they were given from dry dredgers. So really, their contributions to paleontology cannot be overstated. All right, so to conclude here, avocational paleontologists have made immense progress across paleobiology, and that is in all fields, dinosaur paleobiology, paleobotany, microfossils, but specifically talking about echinoderm paleontology, we have really benefited from these collaborations and finding more and beautifully preserved specimens that have been able to change a whole host of what we thought we knew about these organisms. So developing working relationships with career and avocational paleontologists strengthens scientific advancement, and it benefits all parties. And I strongly encourage you, if you live near a fossil group, or even if you don't, please reach out and see if you can make some new collaborations. Reach out with a university if you're an avocational paleontologist yourself. These collaborations really do benefit everyone involved. With that, thank you very much. I appreciate you listening.